Yes.
Okay, welcome to all of you to this session of the seminar in PDEs and applied mathematics that we are celebrating since 2020. Today we have the honor and pleasure to have with us Professor Suncica Canic, a magnificent and experienced mathematician from USA. She earned her PhD in 1992 in nonlinear hyperbolic conservation laws from the State University of New York. In 1999, she joined the University of Houston, where she began collaborating with medical specialists at the Texas Medical Center on problems related to cardiovascular treatment and diagnosis. Her research achievements have earned numerous accolades, including the National Science Foundation Distinguished Mathematical and Physical Sciences Lecture Award in 2007, the US Congressional Recognition for Top Women in Technology in 2006, and the Esther Farfel Award, the University of Houston's highest honor, in 2018. Professor Canic was also elected a Fellow of the Society for Industrial and Applied Mathematics in 2014 for her contributions to the modeling and analysis of partial differential equations motivated by applications in the life sciences, and became a Fellow of the American Mathematical Society in 2020 for her distinguished contributions to mathematics. In 2018, Professor Canic joined the mathematics department at the University of California, Berkeley, where she is currently a full professor. In 2024, she was awarded the Association for Women in Mathematics Sonia Kowalewski Lecture Prize and elected as a corresponding member of the Croatian Academy of Sciences and Arts. Her research focuses on the analysis and numerical simulation of partial differential equations with applications in medicine, active materials, and mathematical physics. Moreover, her work has influenced the design of a stent for bioartificial aortic valve placement developed by a private consortium in Houston and a second-generation bioartificial pancreas currently under development at the Biodesign Laboratory at UC San Francisco. So thank you very much, Sunny, for having accepted our invitation to give this talk, and you can start when you want.
Thank you very much. Good morning from Northern California. Thank you for the nice introduction and for the invitation. I will be talking today about some mathematical aspects of fluid poroelastic structure interaction that we have developed after starting to work with my collaborator Roy at UCSF on the design of a bioartificial pancreas. So the plan for today's talk is to give you a brief biomedical background, then talk about the mathematical model of fluid structure interaction that we designed, and then show the existence result and the main ideas behind the proof of the existence, and then numerical results and how they influenced the design of a second generation bioartificial pancreas. So bioartificial pancreas, which is shown here, this orange banana-shaped organ, is a glandular organ whose main function is the production of insulin to regulate the blood sugar levels. Malfunction of the pancreas is related to a collection of diseases which is known as diabetes, which is a condition that is characterized by insufficient production of insulin, which can be deadly if it is left untreated due to hyperglycemia. So there are different types of treatments for diabetic patients, but the most severe patients have to undergo transplantation therapy, which entails infusing the healthy pancreatic cells from a donor into the liver of a patient, where a particular type of pancreatic cells called beta cells start producing insulin. But as with all transplantation therapy, there is a significant drawback, which is that the patient's own immune cells recognize the transplanted cells as a foreign object and start attacking it. And so those patients are typically on a lifelong immunosuppressant therapy, which can have potentially severe side effects. So to deal with this issue, a group around Chouveau-Roy, who is again the director of the UCSF's Biodesign Lab, they have been working on the design of a bioartificial pancreas, which would be implanted into the patient's arm. This is the device, and connected to, let me move this Zoom thing to the other side. Hang on. Oh. And connected to the patient's vasculature on one end to, let's say, the radial artery in the arm, and on the other end to a vein. This generates a pressure gradient, pressure difference that drives the flow through this tube, the anastomosis graft, and also a part of the fluid, blood flow, namely blood plasma, then filtrates, goes through the bioartificial pancreas. Which consists of transplanted, healthy transplanted cells that are transplanted into a biocompatible hydrogel, which is a poroelastic medium, a sponge. And the cells in this poroelastic medium are then encapsulated between two semi-permeable poroelastic membranes that are especially designed, and this is one of the expertises of Chouveau-Roy's lab. They are especially designed to allow the passage of oxygen and nutrients to the cells, the food, and insulin away from the cells to the patient's cardiovascular system. While protecting the transplanted cells, namely the membranes exclude the patient's immune cells, the cytotoxic cytokines, from passing through the membranes and attacking the organ. One of the key challenges in the design and in the use of this bioartificial pancreas in patients is to maintain the survival of transplanted cells for extended periods of time. And this is a challenge in all bioartificial organ design, namely one would like to provide sufficient oxygen and nutrients so that these cells can live not just for weeks or months, but for years. And so the current approach was to, so originally actually the device was implanted in a pig, and the hope was that entirely due to diffusion of oxygen in the pig's body, the cells would be getting sufficient oxygen and nutrient supply. This pretty quickly showed not to be the case. And so they connected the device to the pig's vasculature, which still was not sufficient. It improved the flow of oxygen through blood through the device, but this was still not sufficient. To enhance oxygen supply to the cells in that poroelastic medium containing the cells, the group around Chouveau-Roy and some other groups have drilled these vertical channels to enhance advection-based oxygen supply to the cells. This also turns out, while improving oxygen supply to the cells is not sufficient, because now the cells can live perhaps for several weeks, but this is still obviously not sufficient. So when we were asked to collaborate with them, one of the questions was to understand what would be the optimal, as they say, architecture of a hydrogel, namely what kind of ultrafiltrate channels can one drill or design inside the hydrogel chemically, so that the concentration of oxygen in the hydrogel would be as close to uniform as possible and above the minimum concentration above which cells can function collectively as an organ. And in general, we were asked to design a multi-scale mathematical model that would describe not only the flow through the poroelastic hydrogel, but also the flow through the anastomosis graft, and the flow, the interaction between the blood flow in the graft and this multi-layered poroelastic medium, which consists of a membrane at the bottom, a thick poroelastic medium containing the cells in the middle, and another membrane at the top. And so at this point, indeed, we have a first version of the multi-scale model that uses fluid structure interaction simulations to describe the flow of blood through the anastomosis graft and through this multi-layered poroelastic medium at the macro scale. Using the fluid information from the velocity of fluid, blood, we then feed that into a advection reaction diffusion solver for oxygen concentration. Oxygen is calculated everywhere in the graft and in the poroelastic medium. But to be able to distinguish between different microscale structures of that poroelastic medium, whether it has channels drilled through it or not, we had to also include the microscale simulations that would inform us of the macro scale parameters that we need to study the flow through the poroelastic medium at the macro scale, such as the permeability coefficients that describe the pore structure of the poroelastic hydrogel. So this is our poroelastic hydrogel simulation with a 2D slice, which is shown here. This is a 2D slice of a hydrogel of the poroelastic medium, which is randomly generated. Colors show magnitude of velocity. And this one is the hydrogel with the vertically drilled ultrafiltrate channels. And then we also included convolution neural networks approaches to generate that permeability tensor that I'm going to show you where it appears in the model in just a second. But the macro scale parameter based on the microscale structure of each new hydrogel, that information is obtained from imaging of a given hydrogel or just synthetically generated using numerical simulations. We then train the convolution neural network on a bunch of synthetic data to be able to offline, to be able to quickly give us information about the permeability tensor for every new image of a hydrogel that is used, that will be used in the design of that bioartificial pancreas. At the micro scale, the simulations that generated that fluid velocity that I showed you here were obtained using the smoothed particle hydrodynamics approaches that are rather expensive, and that is why this approach, where this thing is calculated offline, is and obtained, and then just the coefficient or tensor in our case used in the macro scale simulations is much more efficient. But in today's talk, I would like to focus only on this part, which is the most interesting mathematically, namely the interaction between the flow of a free fluid, not constricted to a poroelastic medium, so flow in the tube, interacting with this multi-layered poroelastic structure. And in particular, we are going to focus on the domain which is in this purple box. So we have the flow through the anastomosis graft. Here it goes from left to right. Here from right to left, but okay. Then we have the poroelastic plate or membrane separating that free flow from the flow in the poroelastic hydrogel. And so this is obviously a multi-physics problem. We used the time-dependent Stokes equations to, or actually in other models, we also used the time-dependent Navier-Stokes equations to study the flow of free fluid of blood in the anastomosis graft. And then we use the classical BO equations for poroelastic media to study the flow in the poroelastic hydrogel. The system of equations consists of two sets of equations. The first one describes the elastic motion of that sponge. So it describes the elastodynamics. And the equation is given in terms of the displacement eta of that poroelastic hydrogel from its reference configuration. Sigma B is the stress tensor, which incorporates the information about the elastic response or elastic energy, elastic properties of the hydrogel. But also, so this is classical, but also it includes this extra term that accounts for the impact of the fluid flow through the hydrogel on the motion elastodynamics of that poroelastic medium. The second equation is a conservation of mass principle, which says that the rate of change of this quantity here given in terms of the fluid pressure passing through the pores and displacement is equal to the divergence of this permeability tensor or permeability coefficient, if you want, times the gradient of the pressure. So this, through Darcy law, is equal to the negative fluid velocity in the poroelastic medium. So this is the rate of change of fluid content in the poroelastic medium. Every subvolume is equal to the divergence of the fluid velocity. And we have taken this matrix or tensor kappa to be either a function of space and the time because we wanted to capture different structures of the hydrogel at the microscale level. So permeability will depend on whether we have vertically drilled channels or not. And also another case that we looked at is a nonlinear model in which K depends on the fluid content because it is well known that hydrogels swell when they are filled with fluid. So this is supposed to capture swelling of the hydrogel. And the plate for the poroelastic membrane or plate, we use the equations that have originally been derived by BO in 1955 from the full BO equations by averaging across the small direction, vertical direction of the plate. So the equations are similar to the BO equations. Here we are using W to denote this displacement. I'm going to copy these equations on the next slide. So W describes the displacement of the middle surface of our plate from its reference configuration, and it has the same form as the BO equations on the previous slides. This captures the elastic energy and so on. And the second equation is conservation of mass principle for the pressure, fluid pressure, where the main difference now is that instead of the full gradient of the pressure here, we only have the derivative in the vertical direction, and that is denoted by the partial derivative with respect to S. Why? Because in 2014, Marciniak, Chokra, and Mikulic rigorously justified these equations mathematically and showed that the dominant flow indeed is in the vertical direction, and that is why Darcy law, for example, reads velocity is equal to the kappa times negative kappa times gradient of the pressure, but only the derivative in the vertical direction. Notice that the first equation is defined only at a codimension 1 surface, the middle surface of the plate, and the second equation is defined on the entire 2D or 3D domain, depends on the context of the problem, omega p. So this is sometimes difficult to deal with, that we are going to I don't know if I'll have time to talk about that, but as a result of the fact that the second equation is defined on a 2D, let's say in this picture domain, we now have two interfaces, omega p minus and omega p plus, between the plate and the free fluid flow and the poroelastic gel on the other side. So as I said, we now have the three different models that describe the behavior of each of the three areas or domains. And what we need to prescribe now is how the fluid flow and behavior of the membrane, for example, impacts the flow in the hydrogel, the flow in the, how do these three models communicate with each other? So the coupling conditions. And the coupling between the flow of fluid, bladder flow in the anastomosis graft and the flow passing through this thin plate is given by these three coupling conditions. They are obtained from both basic physical principles, but also from the weak formulation of the underlying problem. The conditions that are written here will give rise to a reasonable weak formulation and good energy estimates, and that is why they are here. So the first condition is continuity of velocity in the normal direction, which is reasonable between the flow here and here. So this is the normal, prescribing normal component of the velocity. The tangential component of the velocity, fluid velocity here in the anastomosis graft, has a slip. So it slips over the poroelastic plate, and the slip is proportional to the tangential component of the normal stress of the fluid. And the third condition is continuity of pressures effectively here. So pressure inside the plate is equal to the normal component of the normal fluid stress at this interface. At the top surface, we have similar coupling conditions, continuity of, so penetration, continuity of normal components of the fluid velocity. Fluid penetrates from the plate to, for example, to the hydrogel in the normal direction, and continuity of pressures, just like what we had before. But since this interface is elastic, it moves as we already saw. And so we need conditions which will say how. In this model, we have assumed that the middle surface of the plate deforms only in the normal direction, in the vertical direction. That's the displacement W. So the coupling conditions here say that the displacement of the plate is the same as the displacement of the poroelastic hydrogel, the trace of it evaluated at the middle surface. So the two are glued to each other, namely. And the second condition says that the force that drives the elastodynamics of the motion of this plate is due to the jump in the normal components of normal stress coming from either side of the plate. The jump in the normal components of the stress coming from the fluid on one side and poroelastic hydrogel on the other side. So this is the model, and these are the coupling conditions in summary. So the first question that we needed, since this model has not been studied before in literature, and we wanted to solve it, the first question that as mathematicians we asked ourselves was, does this problem even have a solution? And so we wanted to see if there is a solution, if the problem is well posed, if there is a solution, is it unique, and does it depend continuously on the data? And so first we looked at the linearly coupled problem, meaning that all those coupling conditions are evaluated not on the current location of the moving interface, but on the fixed interface. We still calculate the forces acting by the fluid onto the interface that caused the interface to deform. We still calculate the deformation of the interface and of the BO model and so on. But the problem is linearized in such a way that the fluid equations are considered on the fixed domain and the coupling conditions are calculated on a fixed interface. And so this is a linearly coupled problem. In the next stage, I'm going to show we also consider the non-linearly coupled problem. So for the linearly coupled problem, we indeed were able to show that this model has a reasonable energy estimate, namely that the rate of change of the kinetic energy of the fluid in omega F, kinetic energy of the plate, and kinetic energy of the thick BO, hydrogel, plus the rate of change of the total potential energy, which includes the elastic energy of the thick BO, plus the elastic energy of the plate, plus dissipation, which is due to fluid viscosity, due to friction in that slip condition, Biot-Joseph-Safman condition, and dissipation due to the fact that in the poroelastic media, we don't have free fluid flow, but flows through a poroelastic medium constricted, domain. We have dissipation due to the effects of porosity of that medium in the plate and in the thick BO domain. That is bounded by a constant that depends only on the data in the problem. So as I said, we wanted to prove existence of a solution, and just to give you a very quick overview, we have there is a rich literature in studying poroelastic media and porous media flows, from modeling, from studying well posedness of just the BO system, and fluid structure interaction for porous media without considering elasticity. But when we started looking at this, there were only three papers that dealt with fluid structure interaction with poroelastic media, and none of which studied well posedness for this particular multi-layered problem, which arises actually in a lot of applications. And so our main mathematical results, which are the first results in analysis of PDEs related to fluid, to the existence of a weak solution to fluid poroelastic structure interaction, and in this particular case to fluid multi-layered poroelastic structure interaction, are the following. So these are the two following sets of results that we have. The first is, as I said, a linearly coupled problem, and the result says that under some mild assumptions on the constants to be non-negative, and under some reasonable assumptions on the initial data that are consistent with that energy estimate, we showed that if our permeability, so if our permeability, let's say constant, let's say this tensor is a constant KB times the identity matrix, if this is a function of x and t, if this is uniformly bounded away from zero, there exists a weak solution on some time interval from 0 to t. By weak solution, I mean a finite energy solution, a Leray-Hopf type solution for people who are doing Navier-Stokes equations. So finite energy solution on some interval from 0 to t. Then we also looked at the problem in which this permeability tensor is a nonlinear function of fluid content. So we have nonlinearity here. And in this case, we needed to use some compactness arguments a la Aubin-Lyons to prove the existence of a weak solution. But from the first equation, which is hyperbolic, we couldn't get the sufficient regularity of the displacement that we needed. And so we proved the existence of a weak solution for the so-called quasi-static problem, which ignores the inertia of the poroelastic BO domain. And in that case, under the assumption that this nonlinear permeability can be uniformly bounded and bounded away from zero, we get existence of a weaker solution. In the next step, we looked on the right-hand side, we looked at the non-linearly coupled problem. This problem, as this little video shows, takes into account the actual motion of the fluid domain. The coupling conditions are now evaluated at the current location of the moving interface. Both the fluid domain and the BO domain are moving. They are functions of time, and they are not known a priori because the location of the interface and other quantities are that determine those domains are a part of the solution of the problem. So there is a strong geometric nonlinearity in this problem, which presents significant difficulties in the analysis of this problem. Let me show you just a few of the difficulties in these couple of slides, in the next couple of slides. So we, again, our mathematical domain is the moving domain omega f of t, moving BO domain, and an interface which is also moving. We are now using the Navier-Stokes equations for an incompressible fluid to describe the free fluid flow. We are using the same BO poroelastic medium equations in the thick domain, which I'm not going to go through. They are rewritten in terms of some mappings that we don't have time to go through. But the interface now is no longer the same plate that we studied before. Because there isn't a nonlinear version currently of that linearly elastic plate of those linearly elastic plate equations available in the mathematical literature that is rigorously justified. So what we did was we used the so-called reticular plate model, which is just, so this omega is the displacement of the plate from its reference configuration. This is a fourth order derivative acceleration terms. The coefficients in this plate model depend on how many holes the plate has. Details of poroelastic of these types of reticular plates can be found, for example, in this book. But the main point for today's talk is to realize that we have considered that this plate is completely permeable to fluid flow. The flow itself does not directly feel the presence of the plate. It feels the presence of the plate only through its inertia and elastic properties. So the coupling conditions described are the same as what we had before, except that we now have continuity of flow in the vertical direction between the free fluid flow and flow in the BO medium, not the fluid in the plate, because the fluid doesn't see the plate in this way directly. And I'm going to skip this, but we were able to prove that a same type of energy estimate, the total kinetic energy plus the total elastic energy plus dissipation, are bounded, namely they are equal to the total kinetic and elastic energy at time t equal to 0. And so the question was, what can we say about the well-posedness to this problem? Well, there are a lot of new difficulties that we encountered here that have to do with two things. One, the fact that now the BO poroelastic medium is moving, it is no longer considered fixed. And two, there is fluid flow through this structure. If we didn't have poroelastic medium, but if we just had the elastic medium without the fluid flow, there are still some difficulties related to the loss of regularity of the interface between the two, the free fluid flow and the elastic structure, that can, however, be dealt with in certain ways that, well, I can talk about that if one has a reticular plate at the interface. However, the fact that we also have fluid flow passing through this structure gives rise to significant difficulties that I would like to show you now. Namely, for people who are not in this field, I apologize for this slide. You don't have to know all of the details here, but I'm just going to tell you that this is the weak formulation. The red part corresponds to the free fluid flow, the whatever this color is, let's say blue-gray, to the elastodynamics displacement of the plate. This corresponds to the part contributing due to the elastic nature of the thick BO domain, and this is the part that is related to the fluid flow through the thick BO domain. Fine. But if one looks at these three integrals, they are defined on the moving BO domain now. And if one looks at that energy estimate that I showed you several slides ago, one would see that the displacement eta, this is the test function that corresponds to this displacement. The displacement is in h1, L infinity in time, h1 in space. And so this is only in L2. And so to see whether this integral is even defined, one would map the integral from the moving domain onto a fixed domain, omega b. As a result, one would get the Jacobian of the transformation under the integral. The Jacobian is given by the determinant of the identity matrix plus the gradient of the displacement eta. Eta again is in H1. This determinant is only in L infinity L1, so even without the pressure, this integral is not well defined. And so there are three integrals here which are not even well defined for us to be able to even talk about weak solution in terms of finite energy solution. So what to do? So a way to deal with this that we introduced and we did was to introduce the so-called regularized interface or regularized problem, where we convolved the displacement eta, which is the main thing that makes this problem difficult to study. So the displacement of the BO medium, convolve it with a smooth convolve it with a smooth kernel with compact support. This gives rise to smooth displacement eta. And look at this weak formulation and regularize only the smallest number of terms. So use the regularized displacement eta only in the smallest number of terms that give rise to a weak formulation that is consistent to the original weak formulation and gives rise to good energy estimate. So we had to change these three terms and then a couple of more terms to give rise to the finite reasonable energy estimate. And so then we proved the existence of a weak solution to this regularized problem. That was done in this paper and also in the 2024 paper listed here. This is still a difficult problem to study because we still have a non-linearity due to the geometric coupling that we had to deal with. The problem is we are dealing with moving domains, so we had to use new compactness arguments of the form of a generalized Ben-Lyon-Simon compactness lemma that were published in 2019. We obtained the existence of a solution to the regularized problem and then showed that that regularized problem is not something completely out of the blue, but that the regularized problem, the solutions of that problem, constructed in the constructive existence proof, converge as that regularization parameter tends to zero to a classical solution of the original non-regularized problem. When that non-regularized problem has a classical solution. for that higher regularity, we had to assume viscoelasticity in the Biot model. So we showed the so-called weak classical consistency result, which says that those regularized solutions are not something out of the blue, but they actually do converge to the solution of the non-regularized problem when things are more regular. But that depends on the data on the problem in particular. And so there are some references here if anybody's interested that I can share with you. And I don't know if I have time. It's 10:40. The last part I wanted to show some numerical simulations. Do you think that I have, let's say, 2 minutes, or should I stop
Yes,
here?
yes, you can take 2 more minutes. Yes, sure.
Okay.
Yes.
So this is an actual bioartificial pancreas. We have this inlet and outlet. There is a blood flow channel going around like this. And on top of that blood flow channel, there are those semi-permeable membranes through which blood plasma flow enters the poroelastic medium here with drilled vertical channels that contains the cells, and the flow goes then through this gasket and to the patient's vein and the rest of the cardiovascular system. I'm going to skip this. This is an actual implantable bioartificial pancreas implanted in a pig. But so I am going to skip these results and get to the point where we actually then looked at that poroelastic medium itself with drilled channels and calculated the velocity. Here are the streamlines of the velocity. And then also calculated the corresponding concentration, and now flow goes from top to bottom. Red is a high oxygen concentration, and blue, cyan and blue is low oxygen concentration. Obviously, one can see that, especially if I take a 2D slice of what I just showed you, there is high oxygen concentration in the drilled channels and closer to the inlet versus outlet. And so what we then did was to improve the oxygen concentration in that poroelastic medium to be as uniform as possible and close to a constant, above a minimum constant above which these cells can function as an organ collectively. We investigated 3 different scenarios of channels inside that hydrogel, so architectures of hydrogel, of poroelastic medium. The vertical channels that we already mentioned, this was inspired by the branching vessels in the human body, and this was inspired by the epithelial tissue, where these are hexagonal cells and channels in between the hexagonal cells. And then we ran our numerical simulations in for the equivalent scenarios, which means same inlet and outlet data, and also the area of the channels or the volume. In all these 3 cases is the same, constant. And so we showed that indeed the hexagonal distribution of channels provides a uniform concentration almost everywhere in the hydrogel and way above the minimum concentration, which is somewhere in the blue region, above which cells can function. And then we compared those 2D simulations, fluid flow and pressure, to the full 3D simulations and showed a good agreement. And why is this significant? This is my last slide, because actually, because of the recent developments in hydrogel fabrication, they can now chemically actually control hydrogel rheology, the channels. Not exactly as what we recommended here, but they can actually produce hydrogel with roughly that type of architecture, which is also what is being done now in the lab by our collaborator at UCSF. And this is the end. Thank you.
Thank you, Sunny, for your really interesting talk. And yes, I have enjoyed it a lot. We will give the opportunity now to the audience to ask questions or make some comments if they want. I think that usually Juan used to open the micro to the audience. So you are welcome if you want to ask anything to Sunny. Yes, if not, I have some questions. First of all, meanwhile, if anyone wants to ask something, I have found interesting because not only all the effort that you have done or the research group has done to develop all these equations, no, mathematically, and to put them together and to study in different areas, no, different equations, all the conditions in the that they have to verify to be in the boundaries, no? Also the effort that you have done in order to use that to apply in the medical situation, no? And you have explained and showed us the different images, how it is applied in the body. That I didn't know that it is put in the arm, no? The artificial pancreas.
So that would be okay. So I don't know what's wrong with this. Have I lost you? Are you still there?
Yes, we are here. You have frozen.
Okay, am I still sharing this?
No, at this moment, no. Your image has been frozen for some seconds.
Oh, I don't see it like that. Okay, so let me hang on. Let me see.
You are stopped in the
Okay,
image.
it doesn't matter that I don't see what you see, but they still have not implanted it in a patient's arm.
Ah,
This is still being used and tested in animals.
ah,
And things are improving in the sense that um, the time spent, the length of time that the cells can survive now is at the order, as I said earlier, several months. This is still not sufficient.
mhm,
They have not included our hexagonal cells design yet into their poroelastic hydrogel. And one
ah,
of the reasons is funding, which is now being cut, as you know, significantly.
mhm.
And they will have significant consequences for a lot of different people.
Oh, that,
So,
yes. That was another question that you have mentioned the hexagonal channels at the end, and you have shown us the different images for different type of channels. I had one question that maybe you have said, but maybe I haven't understood. Or the hexagonal channels are the best one in some sense, or are,
hang on, let me try to share.
yes, we can see now that you have changed the.
Can you see?
We can see your screen now, and you are trying to find it in
Oh,
the PDF. No?
okay, so let me see if I can. Um, can you? No, now I can. Can you see it?
Yes, we can see there is a main slide, and then below we can see the different slides.
Okay, so let's do that. But what did you say that in the last slide?
Yes, in the last slide you put the different kind of channels with different diagrams.
Ah, yes.
That one, 48, yes. And I was asking if the hexagonal type was the best one in some sense, or
Yes, absolutely. So the hexagonal channels, excuse me, which in a 3D, what we tested was the cross section is a hexagonal, but in the third direction, it's just extruded. It's just, straight channels in this direction. These
Mhm,
channels seem to be, in comparison with the other two geometries, the best ones that are already giving us, even in 3D, oxygen concentration, which is pretty close to uniform oxygen concentration. We do not have a mathematical optimization algorithm to do this.
Mhm.
We know what we would like to optimize. We know what the constraints are. There is a constraint on the volume. So you would like the volume in between the channels to be large enough so that you have a sufficient concentration of those cells that can again function as an organ, and so on. So we know what we would like, and we can impose an optimization problem, but we have not solved it theoretically or computationally.
Yes. So thank you. I don't know if there is someone that wants to ask something in the audience. If not, I think that it has been a really interesting talk, Sunny, because not only because all the mathematical things, I think that those are incredible, that you have done a lot of work, but also because I think that this is encouraging for the young people, no, to try to apply the mathematics in the medical situations and, no, and how can they do so. Congratulations, and I think that a good future, and maybe in the future it can be applied in the human body, no?
Yes.
Yes.
Well, thank you very much,
Thank you, Sunny. You're
and
welcome.
nice meeting you all.
Yes, thank you.
Thank,
Okay,
thank
bye.
you, Professor Suncica, for your excellent talk.
Thank you. Bye.